and a pioneer in the field of autophagy, which was honored yesterday with the Nobel Prize in Physiology or Medicine. So. So for the record, not for Dr. White, but what Dr. White did tell me was that hands down, the person who won the prize was the person who led the field by about a decade, discovering autophagy in yeast and laying all of the groundwork for what we know about autophagy today in mammalian cells and in human cancers, where it's obviously very important. So, thank you for co-sponsoring this event with us, Eileen, and thank you for kicking things off.
Oh, thanks, Stephen. My comments will be brief. I would just say that we've known for decades that the immune system somehow is playing a role in whether you got a tumor or not, and I think there are many early observations to suggest this. Probably the one that sticks in my mind the most is melanoma patients, rare melanoma patients, would have their tumors spontaneously disappear. And these and a number of other observations suggested that something was going wrong with the immune system that was allowing cancer as a disease to progress. And now we know much, much more, and are actually translating these discoveries in the clinic. We know that there are mechanisms that tumors evolve to suppress immune attack by T cells. And there's a whole collection of molecules that the tumors can turn on and off and regulate the immune response of the host. And I think what we've seen is miraculous responses in a subset of patients in the clinic. And I think it's really an open season on what we can do to make the most of modulating the immune system to cure cancer. I think what's of interest of the Cancer Institute of New Jersey is first, clinical trials using checkpoint inhibitors for the benefit of patients, and you'll hear how that is being done. And also from the basic science side, you know, asking questions, okay, if 20% of patients respond to checkpoint inhibition, you know, what about the other 80%? What is the mechanism? What is the mechanism that determines whether someone responds? And so we and my colleagues at the Cancer Institute have projects to address that. So I guess without any further delay, actually, who's addressing? Stephen's introducing the next speaker.
Dr. Stephen Zheng from the Cancer Institute.
All right, welcome. So we're at a very exciting time. You know, now we have a very effective therapy to achieve durable responses in patients, and that's been great. And that also created some fear among cancer pharmacologists that we may be out of business. Because you know, the chemotherapy, the old-fashioned and targeted therapy, where can we be? And you know, I think you know, from today's program, we're going to learn that you know, I just want to follow up Eileen's you know, perspective that you know, this is effective therapy, but you know, there's still a lot of work to be done, and there are quite a few patients they're not responsive. So there are going to be enormous opportunities for targeted therapy, targeting for small molecules to target the you know, intracellular targets in the immune system as well as in cancer cells, and combining the existing therapies, chemotherapies, and targeted therapies with immune therapies to really enhance the outcome. So we're fortunate to have Dr. Hunt from Bristol Myers, who is an expert in this area, give us, overview in this area. So, and again, without further ado, I have Gail to give us some perspective from PDB and
Thank you, Steve. Yeah, so I'm Gail Ferstandig Arnold. I'm the Associate Director at the Institute for Quantitative Biomedicine under the leadership of its founding director, Stephen Burley. What I wanted to do is introduce you to the Institute. It's only been around for 15 months. It's a virtual institute. And our goal is really to complement other programs going on here at Rutgers and to figure out ways to really meaningfully partner with people in industry. And so we've set up some initiatives, and I'd just like to introduce them briefly before we go on to our talks. So the Institute basically brings basic and applied researchers together with clinicians, and the idea is that if we come together, we can actually tackle some of the grand challenges in biomedical research and bringing to bear quantitative tools that can be applied toward the life sciences and biomedical sciences. And we have a graduate program within the Institute that parallels these goals. And so we're interested in training students to be at this interface between the quantitative sciences and biology and medicine. And so there are a few things to share. So basically, the Institute is dedicated to serving as a hub, if you will, to promote collaborative efforts. And so for group leaders, whether at Rutgers or in industry, we're interested in having people come together who share common interests and would like to have conversations about what there is to do in various areas of research. And to date, we've started 2 research working groups, one in cancer genomics and proteomics, co-chaired by Sridhar Ganesan, one of our speakers today, and Stephen Burley also, who will speak today, and one on antimicrobial resistance, co-chaired by David Perlin at the Public Health Research Institute in Newark, along with Stephen. And we'll be adding research working groups. And in fact, it would be wonderful if a research working group develops out of today's gathering, if people find that there are common interests that could be shared and further developed. And we're also inviting faculty, and this could be in industry as well, to join the graduate program faculty of our graduate program to help create a program that's really responsive to what the graduate students are going to be facing when they graduate. And as far as students go, we have a joint Ph.D. program which enables students who are studying in one of the traditional areas to complement that with an element of interdisciplinary study. And the idea is just to broaden the education and training of students so that they can graduate and have conversations with group leaders in different areas and really be responsive to being able to sit around a table and come up with new things. So, and in this program, students actually follow all the rules of their primary program, but we add an element of sort of customized training to complement their current education and their goals to help bridge them so that they will be in a position to have this more interdisciplinary education. And the additional training usually fits in very nicely without adding any time to degree. And another opportunity available to all people in this room, really all people at Rutgers, people in industry, is that we have boot camps and special programs. And coming up this January, right after the holidays and right before the spring semester here at Rutgers, we're going to have an immersive 2-week experience focusing on cryo-electron microscopy for single particles. And this coincides with the arrival of a cryo-electron microscope, which sits almost directly below us here and will be available to people both at Rutgers and in industry. And so the boot camp will basically be consist of morning lectures, which are open to everybody, and then the afternoons will be more hands-on, and so that will be for registered participants. The first week will focus on sample preparation and collection of images, and that will be restricted to the Rutgers community, but week 2 will open up to people in industry as well, and that will be a week to analyze the images, to actually process the data, to visualize, to validate, and to deposit new structures into the protein data bank. And I should say, for those of you who aren't familiar with cryo-electron microscopy, it's really going to advance the field. People are determining structures of extremely large, complex cellular machines with direct imaging. And so one doesn't have to grow crystals, and in some cases, one doesn't even have to purify the samples. You can just look at an entire cell and focus in different areas. So it's a very exciting area, and I should say, for those who are Rutgers students, you can take it as a 2-credit course, and we set it up to mirror the real world in that we'll have teams of people working on structures of different proteins, and we're going to set it up so people are forced to collaborate. They're going to be interdisciplinary teams. So mathematicians will be talking with biologists and chemists and computer scientists, and together they're going to have to figure out how to determine the structure of their protein, what's the story of their protein, how does it work, et cetera. And nobody's going to have enough time to do it by themselves, so they're going to have to divide and conquer and teach one another. And in the end, they do a final presentation. And we also include networking and career mentoring so people feel more comfortable as they graduate actually looking for their next positions. And in fact, some of you in the audience have participated in this networking experience from industry, which has been wonderful for the students. So I just want to take this opportunity to say thanks from all of us here to the sponsors, Bristol Myers Squibb and the Institute for Quantitative Biomedicine and the Rutgers Cancer Institute of New Jersey, and to all of you for being here. So I will now welcome Ned Lattime, Deputy Director at the Cancer Institute of New Jersey, Associate Director for Education and Training, Professor of Surgery, Director of Surgical Oncology Research, and Professor of Molecular Genetics, Microbiology, and Immunology. So Ned is going to talk about the tumor microenvironment and immune surveillance.
Thank you. Good afternoon, everybody. I'm probably going to talk the least about checkpoint inhibitors of anybody in the afternoon. But what I'd really like to do is to set the stage by describing some of the studies that we've done focused on the tumor microenvironment. Okay, where are we here? Here we are. So that's the requisite disclosure slide. Okay. Eileen mentioned in her introductory remarks that we've all known for ages that the immune system must be important. Some of us have been studying it a little longer than others. But you really know you've arrived when the Hanahan and Weinberg Hallmarks of Cancer puts in immunology. It wasn't in the original set. So the overall goal of what I want to talk about this afternoon is our overall goal in the whole area is to develop effective and systemic tumor-specific immunity. Cancer is a systemic disease, and this is critically important. We're going to hear a lot this afternoon about targetable antigens that are required. We're going to talk about antigen presentation, and I'm going to really focus on what goes on in the tumor microenvironment. And then the next steps, including the checkpoint inhibitors, we're going to hear a lot from this afternoon. So what I'd like to do is talk about how we've interrogated the tumor microenvironment to try to identify mechanistic targets, how to manipulate the environment, and when we've settled on or we've identified a potential therapy, then how we move and transition to clinical trials. So this could be any immune response. It happens to be an immune response to cancer. It's in steps. Basically, you need antigen. In the case of tumor, it's tumor antigen released from the tumor. It's taken up by antigen-presenting cells. In our case, we're going to talk about dendritic cells. That along with danger signals, if present, activates the antigen-presenting cells. It travels off to the lymph node, activates T cells, a variety of types. Those T cells infiltrate the tumor, and ideally will have an anti-tumor effect. In addition to the primary tumor, importantly, will have an anti-tumor effect in metastatic cells or micrometastasis. Okay? This is just a schematic of what happens, the normal immune response, but what happens when you put in tumor. And this is limited in what you'll see, thanks to my last slide, a much larger potential catalog of factors. But this is the normal immune response. Antigen-presenting cells take up antigen, they mature and start a T cell cascade. Adding the tumor, what we and others have found over the years is the tumor has a potential of co-opting normal immune regulatory pathways. I'm going to talk a lot about IL-10. It's central here. It blocks this maturation step, which is critical. It can, along with TGF-beta and other tumor-associated factors, can dramatically shift the kind of an immune response one gets. I'm going to talk about two preclinical models that we've done a fair amount of work in. The first is MB49 bladder cancer. These are both transplantable models, although this came from a genetic mouse. This was provided by Ian Summerhays. It's normal bladder urothelial cells that have been mutagenized with dimethylbenzantrazine. It happens to be a male mouse, which I won't get into. These cells, when you transplant them either in the bladder or subcutaneously, they grow progressively. As you'll see, they don't induce a systemic immune response, although they have significant antigens, and they kill the mouse. Thanks to Liz Simpson, who has spent decades on studying transplantation immunology, we know what the dominant class 1 epitope is that we've been able to make a vaccine to. This would stimulate cytotoxic T cells. We know what the dominant class 2 MHC antigen peptide is. It's encoded by the DBY gene. Both of these are on the Y chromosome and will stimulate CD4 amplifier cells. As I said, when this tumor is put into mice, it fails to induce a systemic immune response, despite having these well-characterized and targetable antigens. We also learned early on that these tumors, and I'll show you a slide, but these tumors produce high levels of interleukin-10. And what we learned subsequently to that is that the tumor itself doesn't make IL-10. It's made in the tumor microenvironment by associated cells. Okay, and I'll get to the other model in a minute. Here's MB49 grows progressively in mice, generates no systemic immune response. Here, growing MB49, there is no cytotoxic T cell function. It grows progressively, as I said, and kills the mice. If we immunize mice to some of the antigens, which I'll get to, we can induce systemic immunity and prevent the tumor from growing. So prophylactically, we can immunize the mouse to the known tumor antigens and prevent the tumor from growing. Although if you just give the mouse the tumor, it doesn't induce an antibody or an immune response at all. This IL-10 interest of ours stems from early on when we began looking at patients with bladder tumors and patients with melanoma. We did biopsies and RT-PCR from about 50 of each. And what we found surprisingly was that in, I think it was 48 out of 50 bladders and 49 out of 50 melanomas, we saw a huge IL-10 signal. We went back to our MB49 model and found the same thing. We also found, which has made what our studies particularly simplified, we found that if we took the cell line MB49, there was no IL-10 message. It didn't produce IL-10. There was no way to generate IL-10 production from the tumor. So what we've done is to look at the tumor microenvironment to find out in part, not terribly interesting, but where the IL-10 is coming from. Okay, but it did allow us to use an IL-10 knockout model, because once demonstrating that the tumor doesn't make IL-10, then by using an IL-10 knockout mouse, we have the same tumor, we have the same antigen, but we change the microenvironment. And what we found here is in the normal mice, just about all the mice who are transplanted with tumor die, and we rescue about half of them in IL-10 knockout mice. So IL-10 does have a significant impact. We went back to our chart and asked, where is IL-10 likely to have an impact? And we focused on this early step. It's been known for some time that this maturation step is key to activating effective cytotoxic and amplifying cells. That in the absence of this maturation step, and I think there's another cartoon, what happens is you generate suppressive populations, T regulatory cell populations. And so that was sort of the start of what we were studying. In studies done by Arvin Yang, who an MD Ph.D. student in the lab, who's now at Bristol Myers actually, we wanted to interrogate what happened to that key antigen presenting step, the dendritic cell step. So isolating dendritic cells from MB49 mice, we found that they were substantially hindered in their immune function, in their ability to stimulate an interferon responses, which would be the hallmark of an effective effector population. And if we took the dendritic cells from IL-10 knockout mice, they were fine. They were completely normal and inducing a response. So what we found was that the IL-10 shifted through, we believe, affecting the maturation of antigen-presenting cells, shifted to the inducing a suppressive versus a suppressive population and response, as opposed to an effective effector response. This just shows this in another schematic. What's clear is under the influence of IL-10 to other tumor-associated proteins, TGF-beta and VEGF, that you end up not inducing an effective response, you end up inducing a regulatory T cell response. And that led us to look to the next step at regulatory T cells. Regulatory T cells are one of the two principal suppressive populations that have been identified in tumors, in solid tumors. Here, using two markers for a number of markers for T regulatory cells, what we found within the tumor was a large expansion of T regulatory cells. This is a little misleading that it is so high because the markers that we're looking at are also on effector cytotoxic cells or effector helper cells. However, they're overexpressed at a higher level in regulatory T cells. That allowed us to isolate those cells and look for a particular function. And what we found, again, coming from the microenvironment, what we found in tumor in particular was a preponderance of suppressive populations, suppressive T regulatory cells that blocked indicator responses here. Again, taking advantage of work done by Liz Simpson, we found that these T regulatory cell populations, using IL-10 or TGF-beta as a readout, were tumor specific. They were specific for the tumor antigen that on the Y chromosome. So here we had lack of antigen presenting cell activation. Here we had enhanced levels of T regulatory suppressive populations. We took advantage of having an antibody that would significantly reduce or eliminate T regulatory cells in the mouse. Unfortunately, there isn't the equivalent in man. And so what we found is that if we could eliminate the T regulatory cells or substantially knock them down by treating with this anti-CD25 antibody, and then interrogate what happens in the mouse. This is the slide I showed you before with an added line. Basically what we found was looking here at the tumor growth, here's the growth of the tumor, here's the growth in the gray triangles, diamonds of mice immunized before we gave the tumor. So we can clearly induce an immune response. And here's tumor growth when we got rid of the T regulatory cells. So eliminating the T regulatory population in these mice allows an immune response to be generated and blocks and generates substantial amounts of immune response and blocks tumor growth. So appropriately modulated, we can have a significant impact on the growth of the tumor and the induction of immune response. The key thing here, however, is this is prophylactic. This is before we've given the mouse the tumor. So if we know that if we pre-immunize to appropriate antigens, we can block tumor growth. If we eliminate the suppressive population, we give the mouse the tumor. The mouse is now able to generate a substantial immune response to the dominant antigens that we'd already identified and eliminate the tumor. Problem is, and very few of us who are in the clinical area are dealing with pre-treatment and prophylactic treatment of tumors. What we're really interested in is established tumors. And we took advantage of another reagent that we were able to produce. We made tetramers, again, taking advantage of knowing the dominant antigen that would identify cytotoxic cells, the other arm of the positive response. We made peptide, we made tetramers, and what that allowed us to do was to identify individual tumor-specific T cells that are recognized through flow cytometry that recognize the dominant antigen. Again, this is work that was also done by Arvin Yang a long time ago. Basically, if we use these tetramers, what we find is if we look in the draining lymph node or if we look in tumor, we find a substantial, although we were not able to see a response systemically, but in the draining lymph node, we find a substantial level of tumor-specific CD8 positive T cells. We know they're CD8 positive effector T cells. And if we take these cells out, stimulate them in culture, what we find is that we found a substantial, a very high level of activity of the cytotoxic cells. They have the capability, if removed, stimulated, and they're in the tumor, to substantially kill the tumor. and these are obviously in tumor-bearing mice, and we don't find these systemically in the spleen. And if we try to isolate cells from the spleen, we don't see any cytotoxic activity. So what's happening is within the tumor microenvironment, we are generating a tumor-specific cytotoxic population under the influence, we believe, of the T regulatory cells, unless we get rid of them. But it's not systemic. So what we took advantage of, knowing the genes that encoded the dominant epitopes, class 1 and class 2, we made a series of vaccinia-based vaccines. So we took the DBY gene, we took the UTY gene, we encoded it in vaccinia. Now we have the ability to immunize and potentially expand those two populations. Here, if we're looking at specific lysis, okay, and here there's no immune response in an MB49 mouse. There's little bit of immune response, not much of an immune response in, again, this is tumor-bearing mice, very little immune response here. But if we immunize, what we found was that if we immunize to the cells that are the DBY class 2 amplifier cell population, we can see a substantial enhancement in lysis. In particular, if we mix either of these with eliminating the T regulatory cells. So we still have to primarily eliminate the T regulatory cells. And as an aside, what we found is if we didn't eliminate the T regulatory cells before we immunized into the tumor with the virus that encoded the class 2 gene and would stimulate CD4 helper amplifier cells, what we found is it did a great job of stimulating and amplifying the T regulatory population. So what we found is that this vaccine here, the DBY, actually in just about all our studies, enhanced the growth of the tumor. If we eliminate the T regulatory population before we do that, it's actually the best vaccine we have. So what's happening within the tumor microenvironment, if we immunize with the class 1 cytotoxic T cell driving vaccine, we generate a significant immune response, and we have some activity against the tumor, although it's a very strong tumor. And this is just a summary. This is tumor area. This is survival. What we know is the tumor in the red bar kills the mice very fast. In a therapeutic setting, if we eliminate the T regulatory cells, it doesn't help. It's too late. If we immunize into the tumor with the combination of these two, if we immunize into the tumor, we expand the cells that we've shown with the tetramers are in the tumor, and we generate also a systemic response, not just in the tumor, and we cure all the mice. And the same thing happens when, but if we immunize systemically, okay, we haven't seen any effective cells in the systemic systemic or in the spleen. And so if we immunize systemically, which is where we have always immunized, we have no effect. And all the mice die. But if we get rid of the T regulatory cells and immunize systemically, it unmasks the ability to generate an effective immune response and cure the mice. So what's happening in the tumor microenvironment, in this model, what's happened is that within the microenvironment, kept in check by T regulatory cells, we have the generation of a substantial potentially effective T cell response, but it's not in the circulation. If we do things such as get rid of the T regulatory cells and immunize into the tumor, we can generate an optimal systemic immune response. The second model, so basically this is a T regulatory suppressed population. The second model that we've been studying is from a genetic mouse in FVB mouse. It's called Neu-T. There is the activated HER2 antigen or protein, the one that has the transmembrane, the point mutation of the transmembrane domain. We derived a number of cell lines from the genetic mouse. The one that we're studying, we've called NBT1. It grows progressively in the mammary fat pad. We know what the dominant class 1 epitope is, so we can make a vaccine by encoding the rat Neu gene. The tumor fails to prime systemic T cell response, so it's 2 out of 2 in that. And there's no IL-10 and there's no T regulatory cells. So fortuitously, we've ended up with a model that is highly suppressed by myeloid-derived suppressor cells. And that was luck. So we know that with our vaccinia HER2 that we can generate a substantial systemic and local immune response, whether we give the virus into the mammary fat pad, again, normal mice, or whether we give it systemically. When we grow, however, NBT1 in the mammary fat pad, what we're finding here, and this is just flow cytometry of markers on myeloid-derived suppressor cells, what we're finding here is in NBT1-bearing mice within the spleen, in the systemic population, we see a substantial increase or level of myeloid-derived suppressor cells, and they're really overexpressed in the tumor. So much as we found with the T regulatory cells in the other model, here's a model where we're seeing large amounts of myeloid-derived suppressor cells. We know that if we sort out the myeloid-derived suppressor cells from the rest of the population, that we unmask, as we did with the T regulatory cells in the other model, we unmask substantial ability to generate cytotoxic cells and kill the tumor in vitro. So if we in vitro eliminate those cells, there isn't a really good way to do it in vivo, unfortunately. But we've done the same experiment. So here we again do not have. Here's the Tetramer experiment, the same as we've done with the MB49. We do not have in the spleen, in the systemic population, we do not have an indication in tumor-bearing mice of an immune response against Neu, against the mouse, against the tumor. Okay, we don't see much in the draining lymph node. We don't have tumor here. But what we found is if we immunize, this is just control, if we immunize subcutaneously with our antigen encoding vaccine with GM-CSF to enhance antigen presentation, the control has that also. What we find subcutaneously, as we found before, we don't end up with any systemic immune response. So here's another example of immunizing in the wrong place. When we immunize intratumorally, where we know the effector cells are, we have a substantial level of antigen-specific killer cells, both in the draining lymph nodes and systemically. So same idea. We need to immunize into the tumor. And what happens when we do is, and this is just a plot of myeloid-derived suppressor cells. What happens when we do is, here is tumor-bearing mice with myeloid-derived suppressor cells in this quadrant. Here in the spleen, again, with no treatment, this is just PBS, we see a substantial collection of myeloid-derived suppressor cells. If we immunize with just the control here has a virus that encodes GM-CSF. It's very similar to an oncolytic virus that we developed and that others have developed. Here, interestingly, intratumorally with that control, we see a huge increase in myeloid-derived suppressor cells in the tumor in particular. This actually fits with work that's been published by Bob Vonderheide that talks about the pros and cons of GM-CSF. GM-CSF is known to expand myeloid-derived suppressor cells. If we immunize, now we've added antigen. If we immunize subcutaneously, we don't do much versus control to the myeloid-derived suppressor cells. This actually has the GM in it. But again, if we immunize into the tumor with antigen, the myeloid-derived suppressor cells are dramatically reduced. And as I showed you in the prior slide, here is where we see systemic and intratumoral effector T cells. So the only difference between this subcutaneous or between this control here, intratumoral, and this control here is encoding the tumor antigen in the vaccine. It's not the virus itself, it's not the GM, it's the tumor antigen. And this is just a summary of what we see in these experiments. Here, looking at tumor, if we immunize systemically, it has no effect on anti-tumor activity. The only place that we knock down the tumor here and actually cure all the mice is when we've immunized into the tumor with antigen, not the virus and anything. So here's two examples overcoming two different immune suppressive pathways, where if we can immunize within the tumor, we have the opportunity to induce a systemic immune response, because all of these are end up effects of systemic immune response. Okay? So what we found here in looking at the mouse models are consistent findings. Transplanted tumors grow. Systemic tumor antigen encoding viral vaccine of naive mice, non-tumor mice, this is the prophylactic experiment, always induces an effective killer response and always prevent tumor growth. But once you have the tumor, then the tumor, which by virtue of the fact that we can demonstrate it can be killed by T cells, it can be killed by immunized mice, the tumor has antigens but fails to induce systemic immunity, and here's two separate inhibitory or suppressive pathways. Okay. So having shown that, we really wanted to find an opportunity to move it into man. Because the hypothesis here is that if we can immunize patients with ideally with early disease, and actually ideally with operable disease, that if we can use the tumor as a vaccine target, then what we may be able to do is optimally induce a systemic tumor-specific effective immune response that we haven't been able to do given it with subcutaneous immunization. So taking advantage, so we have an outstanding team at the Cancer Institute. The pancreas group came to talk to me about this possibility. They'd heard me provide some of the mouse data, and I sort of smiled and said, we thought maybe melanoma, we'd done clinical trials in melanoma, but remember, you've got to put the virus into the tumor. How are you going to put it in the pancreas? And the team got together within a year of the mouse data, had written a protocol. We wanted to do the clinical trial in operable pancreas cancer patients. So only about 10 to 15% of patients with pancreas cancer are found when it's operable. And however, it leaves the tumor, the cells spread very early. So even in a completely operated patient with normal margins, the cells are already in the liver. And 75% of those patients develop liver tumor within 2 years. So we felt that the best population to study were patients who were operable. And if we could indeed induce a systemic immune response, it might scavenge the cells that we already knew were in the liver. NCI was not happy with our suggestion because nobody had ever put this kind of poxvirus into a pancreas before. And they suggested very legitimately that what happens if it causes a big antivirus response? What happens if it causes inflammation? Are you going to turn an operable patient into an inoperable patient? Important questions. But what they did, our sort of fallback was, let us treat patients who have just been diagnosed, who are inoperable based on the tumor just leaving the tumor, tumor cells just leaving the tumor, but that haven't been previously treated, and have no measurable metastatic disease. They let us do that, and so what we did was we obtained a poxvirus vaccine from the National Cancer Institute. This is actually a vaccine that had been done in a phase 3 trial of advanced pancreas cancer, and it had been ineffective. We worked with our gastroenterologist, Tamir Ben-Menachem, who did an incredible job. And we basically recapitulated what we had done in the mouse. Patients went to the endo suite. On day 1, they had an endoscopic ultrasound-based scan. We took a fine needle aspirate, about 1 millimeter square of tissue, so that we'd have a thumb tissue. We put a dose of virus into the pancreas. We also gave a dose of another virus into the arm the next day. 2 weeks later, we took them back to the endo suite and repeated the process. So now we have 2 doses into the pancreas. We have 2 small tissue samples. And the patients went on day 2 weeks later to standard of care, which in general at that time was a chemotherapy agent called gemcitabine, which unfortunately doesn't have a lot of activity. Okay. What we saw in these patients, and patient 1 and 5 are, I'll tell you why we've included them, but all the patients were the same. So before we treated, here's the pancreas tumor right there. A year later in this patient, this is all infiltrated with tumor. Same thing with patient 5. You can see a small tumor here, and this is all tumor. So what's happened is, which happens in locally advanced patients, we've seen a dramatic growth of tumor, and unfortunately, all of the patients succumb to the primary tumor growth. We ended up with, I think, 4 or 5 patients who had metastatic disease to start with. Those metastases grew rapidly, and those patients succumbed to the combination. But what was particularly interesting is 8 of our patients had no disease in the liver when we started to treat. So this is a disease when you'd expect almost everybody to have disease in the liver eventually. And what we found is none of the patients who we treated that didn't have liver metastasis, none of them developed metastatic disease. So it fits with our hypothesis that if we are able to induce a systemic anti-tumor effect, then it may be able to scavenge the small metastatic foci. But unfortunately, in our case, if it had an effect on the primary tumor, we really didn't see it. The study wasn't statistically powered really to show any effect. It was a standard phase 1 to look for toxicity. Okay. We've just gotten this data back from colleagues at the NCI. We wanted to know whether or not the immunization regimen induced systemic tumor-specific immune responses. So here are a number of samples where we had enough peripheral blood lymphocytes from the patients, and we saw a little bit of CD4 activity in patient 4 and a small level of activity, CD8 activity in patients 1 and 2. These are pre. This is what we saw in the immunized patients. Again, this is systemic anti-tumor specific responses in the blood in the case of patients. And what we found here is not every patient at least met our assay requirements, but what we found here is patients without metastasis with the 2 asterisks, just all but 1 that we measured, this guy right here, or 2, I'm sorry, developed systemic immunity, but so did patients who presented to the study with metastasis. So we have the potential through immunizing, and we didn't have a control group that we could immunize systemically, but this has been associated with induction of immunity. So we're encouraged. What we're doing now in that area is to develop the follow-on trial. We put a lot of effort into determining whether or not putting pox into the pancreas induced inflammation, induced pancreatitis. We didn't find any. And so now what we're doing is we're designing a trial, which we wanted to do in the first place in operable patients. Because operable patients, as I said, about 75% of them recur within 2 years. So there are micromets already or tumor cells already in the liver. This is just to remind me, we've done some really interesting work with Darren Carpizo. We started in the mouse. We've gone to clinical trial. Now we're taking what we've learned back to the mouse. And with Darren Carpizo's group, we've developed a transplantable tumor model with known tumor antigen that we can put into the pancreas 3 days later, we do the same two-week apart immunizations. And this is a metastatic model. And so now what we can do is to interrogate in the mouse what's happening in the metastasis. What we think is happening is that as the metastatic tumor grows, it sort of recapitulates the primary and is likely going to induce the same kind of suppressive populations and the like, but nobody really knows that. And so we're very excited. In our initial studies, we find basically the same thing that we found in the other mouse models is that intratumoral, but not systemic, intratumoral immunization with the particular antigen has a dramatic effect on survival. So in sum, I showed you a cartoon that showed 3 or 4 or 5 markers that had the potential to be targeted or that would regulate immunology, the immune response. Here, and this is 2013, what we can now show you is all in the red. And here is what we're going to hear a lot about is PD-1, where that acts here, and CTLA-4. PD-1 here, I'm sorry, CTLA-4 here. But here is a growing list of potential targets, both positive and negative, that will affect anti-tumor responses, potentially new checkpoint inhibitors. I said I would say something about checkpoint inhibitors, and that's all I'm saying about checkpoint inhibitors. And here are a variety of already being studied therapeutics that would be active at the individual stages. So finally, didn't show up. So finally, let me thank the people who did the work. In my lab, the preclinical work was done by Christian DeVries, Manny Gabriel, Claude Munkin, Arvin Yang. Clinical collaborators, in particular, Beth Poplin, who was a PI and oversaw the clinical trial that I described, and a number of others. We're working with the Carpizo lab. In this model, we're working with the Grow lab and the White lab and a couple of other models. Thank you. I don't know where we stand time-wise. I think we started at. We have time. Do you want to answer questions now, later, Gail? What do you? Wait. Good. Thank you.